Yeah, so this is really a flash update, and it's meant to share some of our current experience and current challenges we have, and we're very happy to talk with others and learn also, because what we're doing is clearly very similar to what other people who are doing ontology development are experiencing as well. So, so I have a way too long title that you can read, and I'm not going to read it again. So just with some brief background to the OB project, the Ontology for Biomedical Investigations is a user-driven project with the goal of essentially describing the parts of an investigation that are not like covered by Go or the cell ontology or so, but really how an investigation is conducted. So how is the sample processing done, what assays are run, how is the data transformed, etc. So it's an effort since 2007. We've been having weekly phone calls now for over 10 years, which is crazy, and wanted to face-to-face meetings per year. There's no dedicated funding for OB, but rather it's a number of projects that are using OB in different ways, and they're volunteering to the building of the ontology. And so essentially, when you look at OB and say something is missing, that is typically because nobody's working on OB with that subject in mind. It cannot be complete if you don't have somebody contributing who wants to volunteer the time to make that area of investigations complete. It's an open project, and we always have new people joining. And so if you are interested, please do. So this is just an overview of a large number of people who are, in our last roll call said they are currently using OB. So there's over 30 different projects ranging from other ontologies or databases, or something like the Neuroscience Information Framework, which is specifically for neuroscience, doing it a knowledge resource, etc. So in terms of scope, this is the high-level class overview of the OB ontology. So you have on the left the upper-level ontology, which you can pretty much ignore. What we keep on using are essentially things like information content entities, where like data that comes out of an investigation, or a plan specification that would be something like a protocol, say, or a study design. Then we have things like materials, where in OB we cover the stuff in blue, so things like specimens, processed materials, so things that are created on purpose, such as devices, organizations, and then planned processes that are in contrast to biological processes in Go, such as whatever calcium transport, are things that you're actually actively want to do, such as conducting investigation, collecting specimens, etc. And specifically, and as I'll talk about mostly, assays, running an experiment to generate data. In parallel, and that's what I primarily wanted to point out here, is we are importing as much as we can, and that's the whole point of OB, to provide essentially the glue that ties together how you run your investigation and how that then relates to the kind of entities, phenotypes, molecules, organisms, etc., that you're making statements about based on the results of the investigation. And so the assays that we are specifically now working with are essentially, so modeling an experiment in OB. I'll just give one example here on how we do it. Namely, specifically, we are pointing out what are the inputs to an experiment. So in this case, you would have a mouse, you have your syringe and a test tube in which you're collecting blood and then storing that. So essentially, you have a set of inputs and your output, and your output from collecting the specimen is your input to your assay, which in this case, you're running it through a glucometer and getting a datum out. So that's the basic principle for all kinds of processes in OB, but then these participants that are either inputs or outputs also play a specific role or perform a certain function. So for example, the glucometer is not used to, say, store a sample, but it's used to measure something. In this case, it measures the glucose concentration in the sample that you provided it with. So not only do we list the inputs and outputs to a process, but also essentially how do they participate. And that can then get rather more complicated. So for example, I mean, what is the relationship? What is this actually? You have like a concentration value here, and this is the concentration of what? This is the concentration of glucose molecules that are in your sample. So the relationship that you end up trying to model here can be quite complicated. And this diagram that you see here is an outcome of modeling essentially close to 1,000 different assay processes over time, seeing what are the repeating patterns that we are finding over and over. And as you can imagine, we didn't start out with this. We started out with other things and gradually added over time as we experienced problems And as new things were added, we tried to address them. And that's where the problems come in. So we have, as I said, it's a community-driven effort with a number of developers over time, and you have now these ways of describing an experiment that also evolve over time. That means that with a 10-year process, you have a number of things that are no longer consistent when you look at them. Making that even worse, you think that OWL understands that two statements should be equivalent, and typically it doesn't. You have to be a lot more precise than we originally thought. We thought we can infer a bunch of things and then make certain types of definitions equivalent, but that doesn't work as much as you think. OWL is much less generous in allowing to say things in a different way and then retrieving the same things back. So a stupid example is for the use of instruments in an assay, we could say, okay, this is a FACS assay is an assay that has as a specified input a FACS instrument and be done with it. Or you can say that a FACS assay realizes a function that adheres in the FACS instrument. And those two things are nowhere near equivalent. And if you have some assays that use one axiomatization and others that use another one, then you'll miss the part that you're really trying to say the same thing here. And importantly, you can discuss forever which one of these is better, or you can just forget about it. What you need is a convention how you do it so that things are consistent. This is not discussions about right or wrong, but if you have them mixed, you're going to run into problems. As I mentioned, we have the other problem that we have evolving design patterns over time. As we are doing this, we're saying, okay, this might be a better way of modeling things than we thought originally. And then the problem is, how do you test that out? Do you go through your 1,000 assay terms in Prodigy, manually edit the logical definitions, and see, do you get a better axiomatization, meaning you have more satisfactory classification of your assay hierarchy, etc. So this is a nightmare. So, and finally, when you enter by hand Manchester syntax, the way you have a bunch of parentheses and stuff, and you do one wrong and everything is incorrect, it is not really a good and efficient way to have like a large number of logical expressions built by hand in something like Prodigy. So we took assays as our example because that's really the key element of OB. And what we did first is we extracted all logical definitions that we had in OB. And essentially we took all those out and matched them against templates and said, okay, this is one type of logical definition, this is one type, etc., and added them all to a spreadsheet, one column per recurring pattern. And then we find that, like in the example with the FACS experiment, that we have columns that are essentially trying to say the same thing, namely that there's an instrument used in this assay, say, and then you have many of these, and we merge those. Once we have this now, then we wanted to have the ability to modify the logical definition patterns and rebuild OB and then inspect what is really better. So can we change the logical definition and get a more satisfying ISA hierarchy, or can we perform more of the desired queries, of which we have now a bunch of examples. What we're using here is there's a number of tools that do these kind of spreadsheet-based ontology building. The one that we are using is the robot tool that was developed by James Overton, primarily with Chris Mungle and a whole bunch of other people as well. This implements a number of specifications that we had originally identified as desirable for OB, which is the quick term template paper. The funding for this comes from Gene Ontology and an IADB. This is how these templates look like. Essentially, you have your identifier, your term label, then you have annotation properties, such as textual definition, that are listed here, and then you have your object properties, where essentially you have a full Manchester syntax style logical definition, and the percent is then always replaced by the entry in the column. Okay, so this is really where we are at right now. We had originally 40 different types of logical constraints to assays, and this is where these boil down to. So one thing that happens in assays, you have a step for material processing. So imagine as an example for all this, a chip-seq experiment. There you have a chromatin immunoprecipitation step as part of your assay. Then you have a detection technique. In the case of a chip-seq experiment, that's a DNA sequencing assay. A chip-seq experiment, nobody in their right mind would say it is a type of DNA sequencing assay. That is a detection part of the assay itself. Then you have the evaluant, that is the chromatin in the case of a ChIP-seq experiment. An analyte is the kind of thing you're trying to detect in your evaluant, which isn't really relevant here. You might be able to specify what specific devices you're using in a ChIP-seq experiment. There wouldn't be one. You have reagents that you're using. There definitely, but in a ChIP-seq experiment, you always have the antibody which you're using to get to the chromatin. And then you have two different types of the outputs. You have what are you getting back? So in a ChIP-seq experiments, that might be a file with DNA nucleotide sequences. But that is what it is about is really clusters of where whatever your antibody was binding to is enriched for. Then we have three things left that we're not completely sure. We think we can probably split all the inputs that we have into device, reagents, evaluants, and analytes, and objectives we may or may not need. And then we have two remaining other axioms which we kind of have to put somewhere. So essentially now we are modeling all of our assays now in this reduced set of definitions where essentially a given assay, you're just specifying then the class label of whatever analyte, device, evaluant, or so you have, and then we can rebuild the OB assay files. So the assay file is then one example of a number of templates that we're using to generate these standardized following design patterns. OWL files. In addition, we are importing subsets of ontologies such as KB and Go. We're importing entire ontologies where essentially all of it is needed for OBI, merge them with a kind of like a base framework of OB, and then we have the final file. So updating this then now requires just changing the template, and you end up with a new version of OB. So I do have the summary, so I'm actually sticking on time. So we are, as I was trying to point out here, we are moving to a spreadsheet-based ontology editing system with the goal of ensuring that all the logical definitions that we have are consistent, and it allows us to easily update them. One key element of this is also editing a spreadsheet and telling people this is where the device goes is much easier than explaining to them how to change anything in Prodigy. And also this, the ability to test very quickly, just changing the logical axiomatization and rebuilding everything will allow us to rationally decide what is the best design pattern rather than just arguing about preferences. We are doing assays as a first proof of concept, and our goal is obviously to move this to all other classes in OBI. Thank you.
So, oh gosh, that's low. Hello, rock and roll. What happens when the structure of the spreadsheet that you have is wrong for the new axiomatization? So if you come up with a new pattern or you want to change things and the spreadsheet's the wrong shape, how are you going to automate the update and change to that?
That's a good question. So, I mean, there's two different things that could be wrong. So, if the column is missing, it's easy. You just add another column. So, if say I was missing one element here, that shouldn't cause any problems. We have so far avoided a dependency between the columns. I'm not sure if we can keep that up. If we can keep it up, I see no fundamental problems with what you describe. If there are dependencies here, there are definitely issues. Ultimately, we would always consider that the OWL file is the truth, right? So we can say from version 1 to version 2, we change the shape of the spreadsheet, and we can keep track of that. And actually, part of this, we are actually tying back the class definition spec to this was based on template so and so. So for versioning, etc., this should still work. But I'm not saying that we are sure that this is going to be.
Can I sneak in a very quick second question? Just because you mentioned the versioning, how are you tracking the authorial and editorial metadata associated with versioning with respect to the stuff that's in the spreadsheet?
Sorry, can you repeat this?
So you're losing all the versioning information because you can't version spreadsheets in a clear way. Who did this line?
Oh, actually, we can version spreadsheets way better than Ontology. Yeah.
Oh.
So we're essentially working with, a standard diff tool works much nicer because the line is at least in the same place. Having order ensured is easier.
Oh, you do? It's a CSV type thing.
Okay,
yeah.
Take this to the break.
Good point. Thank you.
Is it a very short question?
Yes. Okay.
It's great that you're making it easier for the community to edit these. What thoughts do you have about how to interact with ontologies that don't really want community editing? I mean, how to bring them to the light?
How do we, okay. I'm not aware of that there are ontologies who don't want community editing. I think everybody always wants it, and then they hate what they do, and then that's the problem. I'm hoping that we can avoid that by essentially not allowing them to really edit the ontology, just filling out these fields. But I'd be happy to, yeah.
Okay, so thank you again.